In this video, we'll be introducing the blood, talking a little bit about what its functions are and what it's made up of. The blood is the major transport system of the body. It's circulated throughout the body through the actions of the heart. Well, that's not very anatomically correct. Let's try that again. There we go. Circulated by the actions of the heart. Blood is a substantial part of the body. It makes up approximately 8% of our body weight. Males and females have around 5 liters of blood in them, about 5 to 6 liters on average in males and 4 to 5 liters on average in females. But that's assuming that females on average are smaller in overall body size than males. It's typically a little warmer than what you might think of as your typical body temperature. When you're measuring your body temperature, you're measuring surfaces that are in contact with the outside. But if you were actually measuring blood directly, it would be at about 100 degrees Fahrenheit on average. It is slightly basic with a pH of about 7.4, and it's very viscous, meaning kind of thick and sticky. It's about 5 times more viscous than water. Blood has a number of really vital functions, some of which you're probably pretty familiar with, and some of which you may not have thought of before. The first category of functions is geared up towards transportation. Blood is the major transport system of our body. When we get to the lymphatics, we'll talk about the secondary transport system. But most substances are circulated in the blood. You have oxygen. It's got to get from the lungs to the tissues somehow, and it passes through the blood. You have nutrients. They have to get from your digestive tract to tissues that need them. Transport of waste products to the kidneys, from tissues to the kidneys, or carbon dioxide from tissues to the lungs. And transport of signaling molecules. The hormones circulate in blood. Also critical is maintaining homeostasis. This is another key general function of the blood. Blood flow helps regulate body temperature. You know that your body can control blood flow to the skin. If it'll reduce blood flow to the skin if you're getting cold, and increase blood flow to the skin if you are getting warm, so it helps regulate your body temperature that way. It's also critical for regulating the pH in your tissues. Blood contains a reservoir of bicarbonate molecules. Bicarbonate is a polyatomic ion, HCO3, that acts as a buffer, resisting changes in pH of the blood. The blood also helps regulate fluid volume. When you get thirsty, you are getting that thirst sensation because your blood has become more concentrated, and your brain monitors the blood to determine how much water you need to take in. These last couple of functions, the protection functions, have to do with the functions of the cells, or pseudocells within the blood. The first important function is prevention of blood loss. It may seem a bit circular to say that blood is important because blood prevents blood loss, but it is really critical. Since blood is important for all of these other reasons, it is absolutely critical that the platelets and clotting factors in our blood keep us from losing too much blood. The white blood cells in our blood are also important for the prevention of infection, and this kind of joins in with the transportation function because the blood also transports cells to where they're needed, and it transports white blood cells to a site of infection so that those white blood cells can then gobble up the bacteria or virus that's making us sick. There are also proteins circulating in the fluid component of blood, in plasma, that help prevent infection as well, antibodies and complement in particular. Now, blood is made up of a number of different components. The fluid portion of blood is plasma. Plasma makes up about 55%, so just a little over half of whole blood in a typical healthy individual. This diagram is showing you what whole blood looks like after it has been spun down in a centrifuge. Now, when you spin a fluid in a centrifuge, the densest components sink to the bottom because they're pulled to the bottom by the force of the centrifuge, of the spinning. Plasma stays on top because plasma, it's just watery fluid full of proteins and ions and other things. It's much less dense than the cells, so plasma stays on the top. And you're used to thinking of blood as red, but it's the red blood cells, the erythrocytes, that make blood red. So when you spin it down and separate the erythrocytes from the rest of the blood, the plasma is actually a pale yellowy color. I do want to make the distinction that plasma and serum are not quite the same thing. So plasma does not equal serum. They're not interchangeable. Plasma is just the fluid portion of the blood. Serum is what you have after you take the clotting factors out of plasma. So if you took this whole blood and let it sit on the counter for about half an hour, it'll clot, and the fluid part that you have left is serum. So fluid without the clotting factors. Plasma has the clotting factors. Serum, they've been removed by letting the blood clot. Below the plasma, in this centrifuged whole blood, you have what's called the buffy coat. The buffy coat is this kind of fuzzy, cloudy layer between the plasma and the erythrocytes. And the buffy coat contains the leukocytes, your white blood cells, and the platelets, your clotting specialists. They are more dense than plasma because they're cells or cell fragments, cells in the case of the leukocytes and cell fragments in the case of the platelets, but they're not as dense as the erythrocytes. So when whole blood is spun down, the leukocytes and platelets sit in this little thin layer between plasma and the red blood cells. Note that this is a very small layer, and that's because the leukocytes and platelets are really just a very small percentage of whole blood. Finally, on the bottom, you have the most dense component, the erythrocytes, your red blood cells. This layer is going to be red because the red blood cells are packed with hemoglobin, and that's what gives blood, whole blood, its red color. That's also what makes the erythrocytes so dense. They're very densely packed with hemoglobin, so they spin down to the bottom of the tube, and they make, in a typical healthy individual, just a little bit less than half of your whole blood. Plasma contains a bunch of different substances. It is primarily water. It's over 92% water, in fact, approximately. But it has over 100 identified solutes. 7% of plasma is made up of proteins, right? So 92% water, 7% protein. Albumins are the most abundant protein in the blood. Albumins make up about 60% of plasma proteins. And they are used as transport molecules, meaning that they bind up oftentimes hydrophobic substances and help them circulate through the blood. Albumins also play a really critical role in maintaining fluid balance and in retaining fluid in the blood. They're important for the osmotic pressure of the blood. You also have other transport proteins in the blood. Fibrinogen is the major clotting protein. It's a precursor to the fibrin net that actually makes up a blood clot. You also have immune proteins like antibodies. These are just some of the major proteins. There's others as well, including hormones and enzymes that can vary in concentration depending on what's going on in the body. About 90% of the plasma proteins, including the albumins, are made by the liver, so the health of your liver will be reflected in the blood. If your liver isn't functioning properly, your blood protein levels will decline. You also have a number of other solutes that are not proteins. You have organic nutrients in the blood, so circulating from your digestive tract to your cells, or from your liver to your cells, or from your fat stores to your cells. You have glucose, carbohydrates, amino acids, fatty acids, vitamins. They all need to get to where they're going by passing through the blood. You also have nitrogenous wastes in the blood. These are often on their way to the kidney for processing and excretion. Electrolytes, these are ions present in the blood. Respiratory gases, oxygen and carbon dioxide. And hormones, including protein hormones that fall into this category, but also your steroid and thyroid hormones circulating around in the blood. There is a constant exchange between plasma and interstitial fluid. The fluids surrounding your cells, your extracellular fluid, also known as interstitial fluid, it's continuous with the plasma, and you have this constant exchange from the capillaries, the really small blood vessels in your body, and the interstitial fluid that the capillaries run through. This is how you get substances like oxygen and carbon dioxide exchanging from blood to cells. Oxygen circulating in the blood can get offloaded into the interstitial fluid and then taken up by the cells. Similarly, only in the opposite direction, carbon dioxide produced in your cells get offloaded into the interstitial fluid and picked up into the blood. So, you might be asking yourself, why doesn't all the fluid in our blood just leak out into the tissues if there's this exchange? That's where the albumins come in. Albumins are these big globular proteins. They're too big to leave the capillaries. They can't get out. And since the albumin stays in the capillaries, they exert an osmotic pressure that kind of sucks water in. Now there's proteins in the interstitial fluid as well, so the albumin doesn't take all the water in, but there's a balance between the albumin pulling water into the capillaries and the proteins in the interstitial fluid pulling water out of the capillaries. And this force that the albumins exert, holding on to the water in the blood, is called osmotic pressure. Osmotic pressure is something that we'll come back to over and over. It's really critical when we talk about kidney function. When we're talking about osmotic pressure, we're of course talking about osmosis. This is definitely worth spending some time on if you're a little fuzzy on osmosis. We'll just spend a couple of minutes on it here. Osmosis, remember, is the movement of water across a selectively permeable membrane. And it depends on the permeability of the barrier to both water and solutes. So osmosis only happens if the membrane is permeable to water and impermeable to solutes. Okay? So water has to be able to cross the membrane, but the solutes can't. And the blood vessel walls meet these criteria. They're permeable to water, but they're impermeable to large proteins like albumin. So the blood vessel wall meets these criteria for osmosis to happen. Now remember, the direction of movement depends on the number of total solute particles, right? Osmosis depends on the osmolarity of a solution, which is the total concentration of all solute particles in a solution. This is another way to look at osmosis. We have two tanks containing solutions that are separated by a selectively permeable membrane, so permeable to water, impermeable to solutes. One solution is hypertonic, and remember, hypertonic means this refers to the solution with the higher solute concentration. I'm going to abbreviate solute concentration with those brackets. The hypotonic solution is the solution with a lower solute concentration. Okay? And these are relative, so the hypertonic solution has a higher solute concentration than the hypotonic solution. Since we have these conditions where osmosis can happen, a selectively permeable membrane, and solutions with differing solute concentrations, the hypertonic solution exerts an osmotic pressure. Okay? You can think of osmotic pressure, it's a negative pressure, so osmotic pressure pulls fluids into the hypertonic solution. So the osmotic pressure is pulling fluids, pulling water in that direction. The solutes in the hypertonic solution are sucking water over across the membrane, and the osmotic pressure is that sucking force. So if this were a blood vessel, and this were your interstitial fluid, it would be the plasma proteins, particularly albumin, exerting that sucking force, pulling fluid into the capillaries until you reach an equilibrium. If you look over here on the right, remember osmosis doesn't continue until one side is dry. Osmosis continues until the solute concentrations are balanced, are equal. This is the state of equilibrium. I just want to say one more time, osmosis is a really, really critical concept and worth your while to spend some time reviewing it. Okay, back to the blood. The non-plasma components of blood are collectively referred to as the formed elements, right? So this includes the red blood cells, known as the erythrocytes. It includes the platelets, sometimes also called the thrombocytes. These are the clotting cells, clotting cell fragments. And it includes the white blood cells. These other ones in here collectively are all white blood cells. And the collective term for white blood cells, they're also known as the leukocytes. This is what you see. This image shows you what you see when you look at blood under the microscope. The plasma disappears. You obviously can't really see the fluid. You lose it when you're smearing blood out on a slide. But you do see the erythrocytes. You see the leukocytes. The leukocytes, they tend to look very dark purple because they have a densely staining nucleus. And the platelets, and since the platelets are really just fragments of cells, they tend to be much smaller. They can be very tiny. They can look almost like dust on the slide, right? So erythrocytes, as you can tell from this image, the erythrocytes are what you're going to see the majority of. Remember, they make up almost half your blood volume, so it's mostly erythrocytes, and then you'll see a few scattered leukocytes and platelets. After studying this video, you should be able to answer these questions. We'll go into much more detail about each of the categories of formed elements in future videos.